structure of bacteria. By the end of the video, you will have a clear idea about plasma membrane, glycocalyx, cytoplasm, mesosome, ribosomes, nucleoids. As we know, the structure of bacteria was revealed by electron microscopic studies. Only with the help of electron microscope, we can see the internal structure of bacteria. If we start our study from the outermost layer of bacteria, we would start with the cell wall. As you know, the cell wall is a rigid structure and that provides shape to the cell. In this picture, you can see the yellow colored layer, that yellow portion forms the cell wall. Its main function is to prevent the cell from expanding and bursting. That is the most important function of the cell wall because the bacteria live in hypotonic environment and are likely to take in much water and eventually burst. The cell wall of almost all eubacteria means true bacteria, is made up of peptidoglycan. The peptidoglycan is also known as murine or mucopeptide. This peptidoglycan consists of two components. First one is a peptide portion, and that is composed of amino acid, and a glycan, and that is actually sugar portion. So this peptide portion that is made up of amino acid and the glycan portion that is made up of sugar portion, they together make the cell wall a kind of mesh. Here you can see that meshy like appearance. This is gram positive and this one is gram negative bacteria. Here in both, actually mainly the gram positive bacteria, the cell wall forms just like the next structure of bacterial body is glycocalyx. What is glycocalyx? Glycocalyx, the name, actually what is the meaning of glycocalyx? Glyco means sugar, that we know. Second, calyx means the outer cover, okay, cover. So it's a sugar cover. It is something like a jacket which gives extra protection for the bacteria. And it is an optional structure. That means it is not present in all bacteria. This particular structure is seen only in certain bacteria, like certain pathogenic bacteria. Pathogenic means disease causing pathogenic bacteria, and some marine bacteria, marine bacteria, and some freshwater forms. Okay. In such bacteria, that means the bacteria with glycocalyx, it forms the outermost layer. So normally the outermost layer is cell wall, but the bacteria with glycocalyx, their outermost wall would be the glycocalyx. So the order would be like glycocalyx, first one, outermost layer, then plasma membrane, cell wall, then plasma membrane. This will be the order. So this forms the capsule. It's written capsule. That is the outermost layer. Capsule is a glycocalyx. We are going to discuss about that. The second layer, the inner second layer, that forms the cell wall, and inner to that is the plasma membrane. The glycocalyx is found in two forms. They are found in two forms. First one is slime layer and second one is capsule or capsule. Okay, some people pronounce it as capsule, some others is capsule. Okay, so there are two types of glycocalyx. Some bacteria have slime layer, some other bacteria have capsule. What's the difference between these two? This slime layer is a loose structure. It is a loose jacket around the cell wall. Okay, but in the case of capsule, it's a tight structure. It tight, it is found tightly packed with the cell wall. Got it? It is something like some guys may prefer tight fitting jackets while some others would like loose ones. Similarly, you can compare the loose one to slime layer and a tight fitting foam to the capsule. So here the outermost layer, this outermost layer forms the capsule. You can see here this capsule is found uh, closely, that is found in close contact with what the cell wall. You see slime layer, here you can see the slime layer, the outer green colored structure, and it is formed of a glycoprotein. It's a glycoprotein. The sugar component consists of dextrin, and levo sugar. Okay, dextrin and levo sugar along with its protein portion. So altogether we can call it as glycoprotein. Then, what are the important uses of this glycoprotein? Important functions are, first one is protects cells against desiccation. I told you it is something like a jacket, and this jacket protects cells from desiccation, drying out of the body, drying out of the body. Normally, they are found in aquatic medium. Sometimes, if such conditions come, what happens? It protects the cell against desiccation. Second important function is it prevents the body from loss of nutrition. The same function, that is the outer layer that prevents the loss of nutrition. And third function, from some toxic chemical substances that are present around the body or in the surrounding medium, and it helps to defense against phagocytosis, so engulfing by other organism, other bigger organism, because it forms a tough structure, a rigid structure, so it prevents the organism against phagocytes. Then what about the capsule? It's made up of polysaccharides and some have D-glutamic acid. In the case of slime layer, I told you the slime layer is made up of glycoprotein, but the capsule, that is a polysaccharide, and some bacteria have D-glutamic acid. Glutamic acid. D-glutamic acid is an amino acid, okay, and this particular structure, it is found firmly attached to the cell wall. Here you can see it is firmly attached to the cell wall. The cell wall, that is that green portion forms the cell wall, and outer to that it is what to say, it is in close contact with the cell wall, and it provides virulent property. Virulent means disease causing nature or harmful. It makes the bacteria harmful. That means it gets enough protection. Got it? So it will be resistant to antibiotics and such chemical substances. Okay, then let's see what are the important uses of capsule. This structure helps the bacteria to get resistance against antibiotics in the host cell. It is not that capsulated bacteria is resistant to all antibiotics, but when compared to non-capsulated bacteria, they have higher resistance. There are antibiotics that can kill these forms of bacteria like cephalosporin. 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 Okay, that means these bacteria can be killed by certain strong antibiotics, but normally these bacteria are, compared to other bacteria, they are more resistant to antibiotics. The next important function of this capsule is bacteria get protection from antibodies. Antibodies are immunoglobulins. Immunoglobulins are nothing but proteins. And that immunoglobulins or these antibodies are produced from B cells. Okay, of course, you may be thinking what B cell is. You see, WBC, that is white blood corpuscles, these WBC is divided into two groups. First one is granulocytes, second one is agranulocyte, based upon the presence or absence of granules. These granulocytes are again divided into basophils, eosinophils, and neutrophils. Agranulocytes are divided into monocytes and lymphocytes. Monocytes are spherical, bigger in size. Lymphocyte, they are also spherical but smaller when compared to monocytes. These lymphocytes are divided into two groups, B cells and T cells. B cells, what is the main function of B cells? B cells are actually responsible for the production of antibodies. Got it? T cells, they protect the host against pathogen. For example, in our body, if some pathogens enter our body, what happens? Our body should resist. Okay, for that the T cells play an important role. Okay, so we can compare the T cells with, or we can say they are like policemen. Okay, but the B cells, they are like factories where weapons are manufactured. Here, the antibodies would not get access to the bacteria due to the presence of capsu. So, bacteria with outer capsular protection would get protection against antibodies. Next, we are going to talk about protoplasm of bacteria. Protoplasm is a familiar term, but what do you mean by protoplasm? You see, it's a cell, fungal, bacterial, or plant cell. If we remove the cell wall completely or partially, it's like this. If we remove this completely or partially by chemical means or enzymatic means, then the remaining structure is known as protoplasm. Got it. So the protoplasm consists of plasma membrane, cytoplasm, nucleoid, and non-living inclusions. Plus, then in certain bacteria, plasmids and episomes are also there. So that's about the protoplasm. Let's talk about plasma membrane. Bacterial plasma membrane is unique. And here you can see the plasma membrane, that is the yellow colored structure forms the plasma membrane. This portion, okay, this portion forms the plasma membrane, and it is the outermost layer of protoplasm. Here the cell wall is also given, so don't consider that one. Inner to that, the yellow structure, that forms the plasma membrane. It is living and semi-permeable. It is a living structure, but the cell wall is not a living structure, but this is a living structure, and it is semi-permeable. It allows the movement of certain substances across the membrane. That's why it is called semi-permeable. Functionally, the cell membrane of bacteria resemble mitochondria of eukaryotic cell. That is an important thing, you know. Functionally, the cell membrane of bacteria resemble mitochondria of eukaryotic cell. Let's see what are the important peculiarities, and let's compare mitochondrial mitochondrial membrane and the plasma membrane of bacteria. Mitochondria possess inner and outer membrane. The inner membrane has a number of infoldings. These infoldings are known as cristae. Bacterial cell membrane have folds called mesosomes. Can you see this particular structure? And this particular structure is the infolding of this plasma membrane. Inwardly directed certain foldings of the plasma membrane. In the case of mitochondria, this particular cristae, they play an important role in respiration, that means internal respiration. In the same way, the mesosomes, these mesosomes also play an important role in the respiration of the bacteria. The inner mitochondrial membrane contains the same type of proteins and fatty substances as the bacterial plasma membrane. Another important similarity between the plasma membrane of bacteria and mitochondria is the respiratory electron transport system. Enzymes and succinate dehydrogenase are found associated with the bacterial membrane. You see, the succinate dehydrogenate, it is an enzyme, and electron transport system enzymes. These two types of enzymes are present in the mitochondrial membrane for the production of energy, and the same type of structures are present in the bacterial plasma membrane also. Actually, these two terms, that is electron transport system and succinate dehydrogenase, these two are new for you. I will explain it now. Let's see what is electron transport system. Electron transport system, you can see this picture here. This forms the internal membrane. Got it? This is the mitochondria. The inner membrane forms number of foldings, and here only a small portion of mitochondrial membrane is magnified here. This is the outermost layer, and this forms the inner layer. You see, in the inner layer, you can see number of enzyme complexes arranged one after the other. Number of enzymes are arranged one after the other in the inner membrane. These enzymes are actually meant for energy production. And these enzymes are known as, this series of enzymes are known as electron transport system. It's a system. Got it? So we know that the food that we consume, which undergoes breakdown, and as a result finally we get glucose. But glucose is not the actual final product. The glucose again undergoes breaking down process, and that takes place in the cytoplasm of each cell. The first stage, it occurs in the cytoplasm, and after that the second stage of breaking down process take place within the mitochondria. For that, in the mitochondria, that process first is in the cytoplasm, that process is known as glycolysis. When it enters the remaining component, when it enters the mitochondria, there it again undergoes breaking down, and that process is known as Krebs cycle. Okay, as a result of this breaking down process, certain energy rich compounds are formed. These energy rich compounds are NADH and FADH2, and these two energy rich compound, or we can call as reducing equivalents. Okay, they are reducing powers, and which is filled with enzyme, filled with energy, we can say. So this particular energy rich compounds should be broken down to release energy, and that particular process takes place in this system or electron transport system. You see, from the NADH2, from the NADH or FADH2, the electrons are released, and these electrons travel through this enzyme complexes. The electron travels, it jumps from one complex to another, again from that to another complex, and in this way the enzyme ride, or we can say the riding of electron or jumping of electron. As a result of this riding process or jumping process, energy is released. Got it? It's a process. A number of steps are there in this process. Anyway, we have to study this in detail in the upcoming classes. At that time, we will discuss in detail. But this much is enough for you now. So, in the mitochondria, in the inner membrane, electron transport system is there. Actually, there energy production occurs. Got it? And for that, number of enzyme complexes are there. The first one is known as complex 1, enzyme complex 1. Second is known as enzyme complex 2. Third, enzyme complex 3. In that way, using Roman numerals, they are named. And the second enzyme complex is known as succinyl dehydrogenase. So that succinyl dehydrogenase enzyme is also present in the bacterial plasma membrane, and the electron transport system is also present in the plasma membrane. Got it? So the plasma membrane of bacteria acts as what? Mitochondria of eukaryotic organisms. So plasma membrane of bacteria is same as the mitochondrial inner membrane of higher organisms. Got it? So once more, the food that we consume, which undergo digestion, and we get the smaller unit, that is glucose, and this glucose undergoes breaking down process, and we get glucose is a 6 carbon compound and which undergoes breaking down process, and we get 3 carbon compound called pyruvate. This particular process, this particular breaking down from glucose to pyruvic acid, that takes place in the cytoplasm. Then from there the pyruvic acid enters what is called the mitochondria, where this pyruvic acid undergoes a breaking down process, and that particular process is known as Krebs cycle. And from Krebs cycle, due to this breaking down process, we get certain reducing equivalents, energy rich compounds like bomb, okay, and they are known as NADH and FADH2. These energy compounds release electrons, and these electrons enter the electron transport system, and the jumping of electrons through this electron transport system finally results in the production of ATP molecule, that is adenosine triphosphate, or energy coins. Okay, they are energy coins. Whenever our body requires energy, this ATP molecule is utilized. So that's about the what is called the electron transport system. Then the succinate dehydrogenase, SDH, or that's also known as succinate coenzyme Q reductase, SQR, okay, or respiratory complex 2. And this is an enzyme complex found in many bacterial cells and in the inner mitochondrial membrane. Then it's the only enzyme that participate in both the Krebs cycle and electron transport chain. The next structure is mesosome. Mesosome is also known as chondrioid or chondroid, chondroid, chondroid. Mesosomes are nothing but the internal infoldings of the plasma membrane of bacteria. Got it? And due to the actually, what are the important uses of important peculiarities or uses of these mesosomes? The mesosomes are infoldings, I told you. That's as a result, it increases the surface area of the plasma membrane. As a result, enzymatic activity occurs easily. Got it? They have enzymes of respiration. Just now we discussed about the plasma membrane. The plasma membrane is exactly same as that of mitochondria present in eukaryotic cells. I told you that. So this is nothing but the extension, inward folding of the plasma membrane. So it also works as what? Mitochondria. Okay, so they have enzymes of respiration. Respiration in the sense breakdown of food to release energy. So we can say they also work like mitochondria of eukaryotic cell. Then another function is they play an important role in the DNA replication. DNA replication. We will go into more details while discussing nucleoid. Nucleoid is the genetic material of the bacteria. Got it? So they play an important role in the DNA replication, division of the nucleus. There is no nucleus at all. No, it has an incipient nucleus. We are going to discuss about that. Next, we are going to discuss about nucleoid. Nucleoid is nothing but the nucleus of bacteria. Why it is called nucleoid? Because it does not have the nuclear membrane, the outer covering of the nucleus. So it is known as nucleoid. It's also known as naked DNA. That means it is naked, without nuclear membrane. That means the DNA is exposed. It is immersed in the cytoplasm. That's the reason why it is called as nucleoid or naked DNA. It's also called as incipient nucleus. What's the reason? Because it is not well developed as eukaryotic organism, eukaryotic nucleus. Eukaryotic nucleus is well developed with nuclear membrane. It's different types of proteins, and its structure is entirely different from that of this prokaryotic DNA. So here bacterial DNA or bacterial nucleus is incipient because it is not well developed. In this picture, you can see that the DNA of bacteria It consists of it's a long double-stranded DNA. Okay, it's a long double-stranded DNA molecule repeatedly folded with the help of RNA to form a circular ring. You can see here it's a circular form. In this way it's a circular form, you see, but it is folded again and again. Okay, so then bacterial DNA has no free end. You cannot see here any free end. It is circular in nature. Then they are not associated with histone proteins. Let's see what are histones. I told you in the case of bacteria or in the case of prokaryotic organism, along with DNA, there is no histone. What are histones? You just look at this picture. This is the eukaryotic chromosome. And when we observe through the microscope, if we remove the outermost covering, we get this structure. Okay, and only a small portion of this DNA, a small portion of this chromosome, when we observe through the electron microscope, we get this structure, this beaded coiled structure. Got it? And each bead is known as nucleosome. Nucleosome. Good. This nucleosome, this particular structure, okay, this particular structure is formed by the coiling of double-stranded DNA. You see, this coiling of double-stranded DNA over a particular protein structure that is known as histone. Actually, at the center, 8 histones are there. That is an octamer. Okay, so around that octamer, the double-stranded DNA undergoes coiling. And in what is the why this DNA undergoes coiling around it? It is mainly to pack and order the DNA in a systematic way. So each nucleosome is an octamer of histones wrapped by the DNA double helix. They undergo further coiling or folding to form supercoils like this particular supercoiling, resulting in the formation of the chromosome. You see, this form, the supercoiling results in the formation of the highly condensed chromosome, and finally the result is this particular chromosome. Therefore, nucleosomes are the basic structural unit of chromosome. Each nucleosome forms the basic structure of a unit of chromosome. This model is known as string on beads model. Got it? String on beads model. This is present in eukaryotic higher organisms. And these points are very important from the entrance point of view. Then, let's see what's the relationship between a nucleoid and mesosome in the bacteria. Before that, we have to discuss about the structure of the nucleoid of bacteria. Here you can see this double helical structure, that is the DNA or DNA of bacteria, and this the DNA of the bacteria coils around another type of protein. It is a non-histone proteins, and they coil again and again, and finally we get this structure, this particular structure that will become like this highly coiled structure. So that is nucleoid. Okay, then next, what is the relationship between the mesosome and nucleoid? As we see the circular DNA could be at the center or it could be attached to the mesosome. Sometimes the nucleoid is found at the center of the cell. Sometimes it is found in contact with the mesosome. What's the difference between this? Why it behaves like this? You see, it is found at the center when the cell is not dividing. So this condition is a non-dividing condition. Okay, it indicates its normal state. When the cell is dividing, then the circular DNA is found in contact with the mesosome. It could be the sign of cell division. Why it is found attached to the mesosome? Because mesosome helps in cell division. So that's all. Just recollect the important points that we discussed today. First one is the cell wall. Cell wall, it is a rigid structure and you should study the important functions of cell wall. Second point, outer to the cell wall, glycocalyx is there, and there are two types of glycocalyx, slime layer and capsule. Its important functions also. Then third point, protoplasm. Protoplasm is a cell without cell wall. Okay, so then plasma membrane. This plasma membrane is similar to the inner membrane of mitochondria. Then about mesosomes, that plays a very important role in respiration and nuclear division. Then the last we discussed about nucleoids. It's also known as incipient nucleus because why it is a primitive type of nucleus, okay, and without histone protein. That's all. We will discuss the rest in the coming videos. Bye.